Bismillahirrahmanirrahim. Assalamu alaikum. My name is Bakar Department of Microbiology from Guwahati University of Science and Technology. Today, my presentation topic is hepatitis B virus. So, in this video presentation, we will learn about hepatitis. This is the journey from where I made this whole presentation. The article name is Hepatitis B, the viruses and disease. These are the content of my presentation, including introduction, history, epidemiology, classification, structure, replication, cultivation, pathogenesis, mode of transmission, lab diagnosis, and treatment. So let's start from the beginning introduction. Type B hepatitis is the most widespread and the most important type of viral hepatitis. All of the hepatitis viruses are RNA viruses except HBV, which is DNA virus. HBV causes inflammation of the liver. So as name indicates, this hepatitis is just inflammation of the hepatic cell. In hepatic cell are the liver cell. Hepatocellular carcinoma become the only human cancer which is vaccine preventable. This is a short story of about the history of the hepatitis B virus. So, a scientist, Dr. Brun Samuel Blumberg, discovered the hepatitis B virus in 1967, invented the first hepatitis B vaccine in 1969. In 1976, he won Nobel Prize in Medicine for the discovery of the hepatitis B virus. Epidemiology. It's a ubiquitous virus with global distribution, meaning that it's found everywhere in the world. Most common and serious infectious disease. In fact, one third of the world population, 2 million of the world population has been exposed to HBV. The mortality rate is about 1.3 million, fifth cause of the cancer and tenth cause of the death. Classification. HBV is assigned to a separate family, Hepatidinivirida hepatitis DNA virus, which consists of two genera. Number first, orthohepatidinivirus containing HBV as well as the smooth chuck and ground squirrel hepatitis virus. HBV is hepatidinivirus type 1, which means that the hepatitis B virus is including in hepatidinivirus type. Second is heavy hepatidinivirus containing the beacon duct and gray heron hepatitis virus. Structure. HBV is a spherical double-shelled 42 mm DNA virus with an outer envelope and inner nucleic capsid core, 27 mm in diameter, enclosing the viral genome and the DNA polymerase. HBV is partially double-stranded circular DNA containing two types of antigen. First is surface antigen, S antigen, and core antigen called C antigen. So now this is the simple structure of the hepatitis B virus. This black dot indicates the envelope which contains S antigen, while this blue dot, also called nucleic capsid, uh, contains C antigen. This black dot is the DNA polymerase, which is only one enzyme present with the virus genome, and the genome contains two types of strand, plus strand and minus strand. So now replication. HBV replicates within the hepatocyte. Replication of the viral nucleic acid starts within the hepatocyte nucleus, meaning that within the host cell, the liver cell. Replication resembles that seen in retrovirus. That DNA is synthesized from an RNA template by reverse transcription, just like in HIV vaccine. HBV DNA and protein have also been identified in extrahepatic sites such as bone marrow, spleen, lymph nodes, and circulating lymphocytes. So, other than the liver cells, the HBV virus can also be present like in bone marrow, spleen, lymph node, and circulating lymphocytes. Cultivation. HBV does not grow in any conventional culture system. However, a limited production of the viruses and its proteins can be obtained from several cell lines transcripted with HBV DNA. HBV proteins have been cloned in bacteria and yeast. The chimpanzee is susceptible to experimental infection and can be used as a laboratory model. So the chimpanzee is very similar to that of the human and so many anatomical and physiological functions. That's why we can use chimpanzee as a laboratory model. Pathogenesis. The pathogens mean the generating disease within the host or the development of the disease within the host. So the pathogenesis of hepatitis appears to be immune mediated. So the disease replicate and the hepatocyte reflected in the detection of the viral DNA and C antigen in the nucleus and S antigen in the cytoplasm and at the hepatocyte membrane. During the incubation period, high level of virus are present before the host immune response develops and control the virus. During replication, S antigen and C antigen are also present at the cytoplasmic membrane of the host cell. These antigens induce both B and T cell responses. Damage to the hepatocyte can result from antibody-dependent natural killer cells and cytotoxic T cell action. Hepatocytes carry viral antigen and are subject to antibody-dependent natural killer cell and cytotoxic T cell attack. In the absence of adequate immune response, HBV infection may not cause hepatitis but may lead to carrier state. Therefore, infant and immunodeficient persons are more likely to become asymptomatic carriers following infection. Now, mode of replication. So, how the HBV virus transfers from a person to person. So, there are so many reasons. The first is vertical transmission from mother to child, horizontal transmission from person to person by sharing needles and syringes, blood transfusion from infected person to a patient. Tattoos are body piercing using non-sterile techniques, multiple sex partners, saliva, semen are vaginal secretion, and the most important statement about the hepatitis B virus is hepatitis B virus does not spread through food or water. Shared utensils, coughing, sneezing are through touch. Left diagonal. So how we can identify or how we can confirm the presence of the HBV in a person. So there are so many reasons. The first one is detection of the viral marker. The viral marker are the antigen, the S antigen and C antigen, viral DNA polymerases, polymerase chain reaction, and biochemical tests. So by doing different type of chemical tests, we can confirm the presence of HBV virus. Treatment. So no specific antiviral treatment is available for acute hepatitis B virus infection. Hepatitis B immunoglobulin may be administered within a week of exposure into a newborn infant of hepatitis if hepatitis B virus is antigen positive mother. Interferon alpha alone or in combination with other antiviral agents such as lamivudine and famciclovir has been beneficial in some cases of the chronic hepatitis. Some other antiviral drugs used in the treatment of hepatitis B viruses are anticure, tenofovir, tenofovir, and talbutine. These are the complications. Complications mean that a person encounters two different types of disease uh, uh, subjected by that given disease. So the complication, the first one is cirrhosis or scarring of the liver cancer, liver failure, kidney disease, and blood vessel problem. And last, these are the references. And thank you for